Welcome to this lecture on P elements in Drosophila. Imagine a massive, perfectly organized library of instruction manuals where some pages have a mind of their own, magically cutting and pasting themselves into different books. Today, we will explore these fascinating jumping genes in fruit flies, understand how they cause severe mutations, and see how scientists have harnessed them to revolutionize modern genetic engineering. To begin our journey, we must first understand what a transposable element is. Why does this matter? Because DNA is usually thought of as a stable, fixed blueprint. However, genomes actually contain transposable elements, which are essentially jumping genes. They have the remarkable ability to cut themselves out of one location and paste themselves into a completely different part of the DNA. In the fruit fly, Drosophila melanogaster, the most famous and well-studied of these is the P element. When these elements jump around, they can land right in the middle of an important genetic recipe. This physical disruption can ruin the instructions, leading to severe mutations or altering how nearby genes are expressed. Now that we know what jumping genes do, let us look at how they are built. A P element is a specific type of DNA transposon found naturally in wild fruit flies. Structurally, it is quite elegant. At its core, a complete P element contains a central gene that codes for an enzyme called transposase. You can think of transposase as a pair of molecular scissors responsible for actually moving the element. But how do the scissors know where to cut? The central gene is flanked on both ends by short DNA sequences called terminal inverted repeats, or TIRs. These repeats act as physical handles. The transposase enzyme specifically recognizes and grabs onto these handles to initiate the jump. Understanding the structure naturally leads us to the mechanism of movement itself. Why is this specific movement so disruptive? When the transposase enzyme is produced in the cell, it seeks out and binds to those TIR handles we just discussed. Once attached, it literally cuts the entire P element out of its current location in the chromosome. It then carries this DNA sequence and pastes it into a completely new target site. This is known as a cut and paste mechanism of transposition. Because the element physically removes itself and forces its way into a new location, it leaves behind a broken DNA strand and often disrupts the new gene it lands in, causing significant genetic instability. Before we move on to the consequences of this movement, let us see this mechanism with a concrete visual diagram. At the top of the image, you can see the linear DNA strand containing the central transposase gene, flanked by the pointed terminal inverted repeats. Notice how the purple transposase enzymes bind directly to these repeats. As we follow the arrows downward, the enzymes bring the ends together, looping the DNA and cutting the entire element out. Finally, at the bottom, you see the target site. The transposase enzyme is pasting the excised P element into this brand new DNA location. This clear cut and paste action is exactly what drives the genetic changes we will discuss next. Now that we have seen the mechanism, we can understand the massive biological mystery it caused. Why do we care so much about a jumping gene in a tiny fly? Decades ago, scientists noticed that mating certain wild flies with laboratory flies produced completely sterile offspring with massive mutation rates. They called this catastrophic failure hybrid dysgenesis. Without knowing about P elements, scientists were completely baffled. We now know that unregulated jumping genes caused this chaos. Today, this discovery is incredibly important. Geneticists have hijacked this natural jumping ability, replacing the transposase gene with custom genes. This turns P elements into delivery vehicles, or vectors, allowing scientists to insert new DNA directly into a fly's genome, essentially birthing modern animal genetic engineering. While the biology is fascinating, we can also mathematically model how unstable a fly's DNA will be. This formula comes from a simple linear relationship based on how many jumping genes are present and how often they jump. The formula is m equals n times r times p. Let us break down every symbol. M is the total expected number of new insertion mutations per generation. N stands for the total number of active P elements currently in the fly's genome. The variable R is the baseline rate of transposition, meaning the probability that a single element will jump. Finally, P is the probability that when it jumps, it lands in a functional gene rather than junk DNA. If the number of active elements N increases, the total mutations, M, will increase proportionally. Now that we have our formula, let us see this with a concrete example. We are given a wild male fruit fly with 40 active P elements. The jumping rate is 0.05 per generation, and the probability of hitting a critical gene is 0.10. We are asked to find the expected number of noticeable mutations in a single sperm cell. We chose this formula because it perfectly models the linear relationship of these probabilities. First, we substitute our given values into the equation. We multiply 40 by 0.05, which gives us 2. This means we expect 2 actual jumps to occur in this cell. Finally, we multiply those 2 jumps by the 10% chance of hitting a gene, giving us a final answer of 0.20. Physically, this means there is a 20% chance of a new noticeable mutation occurring, showing exactly why active P elements make DNA highly unstable. Having calculated the mutation rate, we must now ask, why doesn't every wild fly suffer from these mutations? This brings us to the biological rules of inheritance. In nature, wild flies, called P strains, have P elements, but they also produce a repressor protein in their cytoplasm that completely stops the elements from jumping. Laboratory flies, called M strains, lack P elements entirely and therefore also lack the repressor protein. The key to hybrid dysgenesis lies in reproduction. The mother provides the egg, which contains the cytoplasm, so she alone dictates whether the protective repressor is present. The father provides only DNA via his sperm. If he is a wild fly, he introduces the dangerous P elements into the egg. To predict the outcome of mating these different strains, we can translate the biological rules into a logical formula. Conceptually, dysgenesis only happens if the father brings the jumping genes and the mother fails to provide the shield. The formula is D offspring equals P sperm times the quantity 1 minus R egg. Let us define the terms. D offspring is the probability that dysgenesis occurs. P sperm is assigned a value of 1 if the father provides P elements and 0 if he does not. R egg is 1 if the mother provides the repressor in her cytoplasm and 0 if she does not. Notice the term 1 minus R egg. If the mother provides the repressor, R egg is 1, making this entire term 0, which physically means the egg is completely protected regardless of what the father provides. Let us apply this logical formula to our first cross. We are given a mating between a wild P strain male and a lab M strain female, producing 150 offspring. We need to find how many will suffer from dysgenesis. We use our logical model to evaluate the inheritance. The P male provides the jumping genes, so we set P sperm to 1. The M female lacks the repressor in her cytoplasm, so we set R egg to 0. Substituting these into our formula, we get 1 times the quantity 1 minus 0. This simplifies to 1 times 1. Our final answer is 1, which means 100% of the offspring will experience hybrid dysgenesis. Physically, this means all 150 offspring will suffer from severe mutations and likely complete sterility, perfectly explaining the historical mystery. Now, what happens if we flip the parents? Let us look at the reciprocal cross, mating an M strain male with a P strain female. Again, we want to find the probability of dysgenesis. The M male does not have P elements, so we set P sperm to 0. The P female, however, provides the protective repressor in her egg, so we set R egg to 1. Plugging these into our formula, we get 0 times the quantity 1 minus 1. This simplifies to 0 times 0. The final answer is 0, meaning absolutely 0% of the offspring will experience dysgenesis. Physically, this proves that the maternal repressor completely protects the genome. It shows exactly how maternal cytoplasm controls the stability of these jumping genes, keeping the offspring perfectly healthy. We have covered a lot of ground today, so let us summarize the most important ideas. First, P elements are fascinating jumping genes in fruit flies that move using a cut-and-paste mechanism driven by the transposase enzyme. Second, we learned that they are the primary cause of hybrid dysgenesis, causing sterility and mutations, but only when introduced into an egg lacking a maternal repressor. Finally, by understanding their mathematical mutation rates and biological regulation, scientists have been able to harness P elements as powerful delivery vectors. This breakthrough allows us to insert custom genes into flies, making them one of the most important model organisms in modern genetics. You now have a solid understanding of P elements and their incredible impact on biology.